This meeting is being recorded.
Utilizing proton NMR, we can distinguish between compounds that, under utilization of IR or mass spec, would generally be difficult to distinguish between. So, with IR, we would need, different functional groups to be able to recognize different compounds, and MS generally. Molecules like these three shown would have the same types of fragmentation and the same peaks noticed. So it would be challenging to just differentiate between these three compounds utilizing both IR and MS, but with proton NMR, we can look for first, the most easily to differentiate would be the number of signals. So what we see here are three signals will be produced because we have here one type of proton. These are chemically equivalent. So say we did an axis here or split this molecule in half, we could see easily three different environments for these protons. So then we have our second set here, and then these 6 protons would collectively produce the third signal. So again, we see 1, 2, 3 distinct signals here. Then we have within our second molecule, we have another plane of symmetry that can be found. So here we would produce 4 different signals. We have this proton here is distinct by itself. These 2 would produce the second signal. So far we have 1, 2. These 6 protons would be equivalent, so this would produce a third signal, and then this one by itself would produce the fourth signal. Now we have the most symmetry within this molecule here, and we would actually produce only 2 signals. So we would have these 6 protons would produce the first signal, and then we could see these 4 produce a second signal. So it's easiest to distinguish between compounds based on the number of signals, but we can also look at the shift of specific signals and things of that nature to further differentiate when we have different functional groups. But again, it's the number of equivalent protons and the fact that these do not have the same number of equivalent protons that allows us to differentiate here. So overall, in section 15.10, we can look at the 5-step process that can be used to analyze an NMR, specifically a proton NMR spectrum. So step 1, if you are given a chemical formula, you should calculate the hydrogen deficiency index, also known as that degree of unsaturation. So there's a chemical equation that we looked at in our IR videos that can be utilized again to calculate the HDI. Then you want to look at the number of signals. So first is the number of signals. So this tells us how many different protons there are as far as different chemical environments, and then the integration of these signals. So that will tell us the number of protons that give rise to that signal. So are there 2, 3, 5, 7, etc. And then the number of signals tells us how many different types. So different types. And then we know the number of protons by integrating. So after we analyze each individual signal, then we can draw fragments of our compound consistent with each signal. So for example, is it a carbon that's next to an oxygen of an alcohol that's consistent with that signal, for example? Then we can assemble the fragments into a complete structure. So we know that there are certain rules that apply to bonding patterns, such as carbon forms 4 bonds, oxygen likes to form 2 and have 2 lone pairs, etc. And then in our last step, after we assemble our molecule, then we can kind of match up each of the peaks and make sure that all the spectral data is consistent with our proposed structure. So we'll follow this plan when we do our spectral analysis utilizing proton NMR.